We have Mr. Aaron Geiman. Aaron's a case instructor and is going to be covering some curricular opportunities for you in regards to biotechnology. And as you may be aware, biotechnology and agricultural biotechnology is the content area of focus this coming 2020-2021 school year for the center's professional development program. And there'll be more on that tomorrow, I believe. So without further ado, Aaron, take it away.
Hello, everyone. I'm just into a gallery view here so I can see some of you all. As I was introduced, my name is Aaron Geiman. I've been teaching for 23 years in Maryland. And I've been teaching the biotechnology course for the CASE project since its origination several years ago. It's been so many I can't quite remember, but I've enjoyed it a lot. And I was contacted a few weeks ago and by some of your program planners today to discuss with you biotechnology. And so at this session, we're going to go into some of the beginning and introductory pieces of biotech. And then later on at the second session, we're going to get into more of the how-tos. I'm going to share my screen really quick here and bring up my PowerPoint. It's pretty straightforward and fairly basic. While I'm doing this, if you guys have questions and so forth, feel free to post them and I'll try and get to them maybe as we end the PowerPoint. I don't think this session is going to last the complete 45 minutes. I've tried to plan about 10 minutes at the conclusion of the session to handle questions. Okay, so first off, I'd like to start off with, what is biotechnology? How I handle it with my students in my classroom. Generally, my students are going to be in 11th, maybe 12th grade when we take this course. Even so, they hear the words biology and technology and biotechnology a lot, but I like to keep it relatively straightforward and simple, and I kind of go backwards and maybe oversimplify the definition a little bit for my students. And, we talk about the root words, word morphology. Biology is life, and technology is the things we do to make life easier. If we want to write something, we use a pencil, and a pencil is a piece of technology, albeit very, very, very simple technology. It still makes communicating easier and more efficient. So putting those two things together, it's just simply using things from life, processes of life, stuff from Mother Nature to make our life easier and more efficient. That's how I want my kids to regurgitate that information when they're discussing it. And moreover, if they're talking about biotechnology with someone who's not quite as scientifically versed, that might make better sense than some of the definitions that we sometimes find in textbooks and so forth. I would also like to stress that biotechnology, as with all technologies, are simultaneously beneficial and detrimental. And an awareness of that dichotomy is important from the scientific standpoint, so that students understand just because we can do something doesn't mean we should do that, or it doesn't mean that we shouldn't do it because there's a lot of detriments. So they need to be able to identify both sides of that coin. Okay. All right, moving on. So at this session, we're focused on the right-hand box, which I've outlined in red. Our big topics today are going to be, understanding biotechnology from a consumer context, awareness and literacy. And then a beginning biotech type of lesson that I've put together here for you guys. We're really going to focus on the interface of science and society and how that influences people's perception of what is good, what is bad, why they may or may not like biotechnology. We'll talk about why that is important to study because of the influence on policy and legislation. Our elected officials are going to listen to their constituents, and if their constituents are strongly for or against biotechnology, that's going to play out. And ultimately, though, some of those choices are making their way into the home. And 90+% of our folks in the nation are not super ag-related. 2% are actually on the farm raising the crops, but everybody has to eat. And so they're being influenced by information, whether true or false. And it ultimately influences the choices that they make with their purchasing dollar. And so we listen to consumers and they exercise responsibility about biotech. So later at the session at noon, we're going to get into some more around the mastery context, career preparation type things. And so if you're interested, tune back in at 12 o'clock. And so a little biotech history. And I'm going to ask Dr. Seaman if he can come back in real quick here, because I am not able to see the question and answers. While I'm sharing my screen, I'm seeing a minimized window. How do you blow that up so that I can see people's, or am I not able to because I'm projecting my screen?
What specifically are you looking for? See people's video?
Questions and no, the questions and chat. If people have a question, it comes up real quick.
Yeah, to view the chat, you just click on the chat thing, and it should pull up a window that you should be able to see even with share screen. And there are no questions in the Q&A pod at this point.
Okay, because what I'm seeing,
what
I'm seeing is I'm only getting this little tiny thumbnail in the corner of my main screen while I'm showing the shared screen. Okay, well, we'll just keep going on. If something pops up, you can interrupt me. And
I'll keep an eye on both for you.
Wonderful.
So you can focus on. Yep.
I appreciate it. Thank you.
Absolutely.
All right, so back to it. Biotechnology isn't new. If we take the idea that biotechnology is using Mother Nature's strategies for human efficiency and stuff, we can start to see the first biotech coming into play with ancient Egyptians using yeast to make alcohol and bread. They didn't know exactly what was going on. They didn't know what was happening in the molecular or even the submolecular level, but they knew that yeast was working for them. We go forward, another thing, the Chinese were using molds to treat disease. Once again, they were probably feeding people these in the forms of cheese and some other things, and the people were getting better, but they probably truly didn't know what was happening. Louis Pasteur in the 1800s links microbes to fermentation. Koch develops the postulates for infectious disease. And we actually start to see artificial insemination and embryo transfer occurring in rabbits in the 1800s. So now scientists are developing and using some of the methods of Locke and Descartes and everybody to start asking themselves, well, what's happening and how can we methodically find answers so that we can reproduce? 1900s, Fleming discovers penicillin. Watson, Crick, and Franklin discovered DNA molecule. We start to see recombinant insulin come out. Disease-resistant tobacco is one of the first agricultural crops to actually incorporate genetic engineering as we think of it today. Of course, actually just the other day was the anniversary of Dolly the sheep. Flavor Saver tomatoes are the first food crop that's approved for sale. And then, of course, the Supreme Court rules that GMOs can be patented, leading to some of the problems out there in today's world because people don't understand why agriculture and biotechnology companies will not let you harvest and retain your own seed and plant next year. If you've watched the Food Inc. Mockumentary, talk about that with the Black Car Squad. And then finally, here in the current century, we see glowfish released into the pet industry, and they're also used for environmental testing. Of course, Roundup Ready crops are out there. Semen sexing is very, very commercially viable nowadays, especially in the dairy cattle industry. In the last 2 years, CRISPR technology is actually being used in the human race by a Chinese scientist who disappeared for a while, and he's come back, but there's a lot of controversy out there regarding that. And Then the Supreme Court comes back and rules that genes that occur naturally cannot be patented. So if I take a gene from one crop and put it into another without actually altering the gene other than just simply moving it, so I can't really patent that and financially protect myself. So the big picture that I convey with my students as we do timelines, and this is not just timelines in biotech, but generally timelines in any class, is that in ancient man, they may not have known exactly what they were doing, but as time progressed, technology became smaller. They were able to dig deeper and move into the microscopic and molecular levels and even the submolecular levels. And so now we're performing biotechnology at the individual atomic level, where we're changing individual atoms in certain molecules so that we can change their influence and effect and efficiency. So I never have my kids remember all of these things on the timeline, but rather I want them to remember the overarching concept that as time and technology progresses, we're getting smaller and smaller. And I would also have my students conceptualize the idea that the density of technological progress increases, or in other words, the speed increases. We see on this timeline the gap of 4000 BCE to 500 BCE. And then we jump forward only another 2300 years, but then we're jumping only by 100-year increments. We can also see that the number of advances on this timeline become more as each line or each bullet on this list has more things to share. So biology for beginners is a little bit to see that we've been doing it through time and we're getting faster and we're getting more things. Wonderful. So some discussion items here. I'm going to give you some things to think about and things that you can maybe bring forward and teach. I know as a teacher's conference, a teacher's workshop online, you're wanting, what can I do in my classroom? And we're going to get those in a few moments. But I wanted to start sharing some of the current directions to build your awareness. This is a little bit of background building. So why do we use biotech in plants? Well, first, disease and pest resistance. Of course, the mosaic virus was genetically blocked or limited in tobacco many, many years ago. I'm sure many of you have heard of Bt corn. It's Bacillus thuringiensis, a soil-borne bacteria, was producing a chemical that we knew was not enjoyed by the corn borer, and so scientists plucked out the gene from the bacteria, stuck it in corn, and now the corn green matter, the leaf and stalk tissue, produces the chemical that deters the European corn borer. So it produces its own pesticide and therefore resists the pest. And of course, papaya, a very popular tropical fruit, would actually be extinct in today's world if not for genetic engineering to resist the ring spot virus. That would be a crop that would not exist. So, of course, glyphosate resistant crops is something that's really popular. You know, obviously glyphosate is the active ingredient in Roundup, but we see that the patent on that has run away. And so a lot of other crops are being developed that are not just Roundup ready, but resistant to glyphosate as it's being produced and incorporated by other manufacturers. We also might take plant matter and biotechnologically with it to increase its nutritional value, such as high lysine corn used in poultry production, swine production and poultry production. And we might also enhance bananas and certain crops that people in other parts of the world eat to provide for nutritional deficiencies that are inherent or endemic in their area due to soil issues or due to dietary restrictions. The idea of farming with a pH, which means that we're now taking and putting pharmaceutical-like chemicals or pharmaceutical-like items into plants for people to eat so that they can get medicines without actually having to take a medicine pill. Is out there in the world. And so that's sort of a nutritional enhancement. And of course, then efficiency enhancements, which were some of the first things that scientists were looking at, such as the stacked genetics and cereal grains, quadruploid and tetraploid genes put into wheat and barley and stuff like that, or drought tolerance in rice. During the early phases of the CASE biotechnology curriculum development, we were actually at a crop science company working with their crop scientists about biotechnological issues, and they were kind of telling us what we should be teaching students. And they were actually working on isolating a gene from mountain rice or upland rice that would help it withstand conditions of low water level. And they were hoping to harvest that gene and put it into wetland rice to increase wetland rice's tolerance of drought. So instead of 6 days of drought, 6 weeks of drought. And so think about the efficiency of that and the ability of rice to provide food for the world. And so that was something that was really, really big. Of course, you know, we were not allowed to take pictures or anything of that. And that still, I don't think has hit the world yet, but it's really, really cool what they're trying to do there. Biofuels, trying to figure out more efficient ways that plants can develop cellulosic compounds that can be fermented into alcohols to replace fossil fuels. And of course, bioremediation, which refers to using living organisms to fix problems. And a little bit later, I'm going to share with you a couple of product vendors that sell bioremediation kits that's for simulated oil spills. So Sarah Beth, Stack Genetics. Years ago, scientists figured out that if they could stick more of the same genes into a plant, then they would be more productive. So scientists took the genes, trimmed them out, and just stuck them in, and they laid them right beside the previous gene. The genes will find the gene similar to themselves and kind of hang out with it. And in doing so, you might have 4 or 5 copies of that gene right in there. They may not necessarily be in line with the DNA molecule and with the double helix, but rather think about like the short gene laying side by side like books on a shelf. But in doing so, that means that when that gene is activated, they'll be producing that many more times protein is what the single gene would have. So let me give you an analogy for that, Stack Genetics. If I'm running a bakery and I want to make more pies, then I might have a cake assembly line, I might have a donut assembly line, a tart assembly line, and a pie assembly line. And if I want to make more pies, well, I'll just add 3 more pie assembly lines, thus increasing my output. And I might put them right beside the other pie assembly line. And they're not sequential, they're not back to back, because obviously one assembly line is going to assemble a pie from beginning to end. So it's senseless to have them stacked sequentially, but rather they're in parallel. And by doing that, I've just increased the number of pies I make. That's what Stack Genetics. Does that answer your question? I can't quite see you, so if you want to give me a, so wonderful. All right, moving on, animals. Animals is where my passion is, because my degree is in animal science and agricultural education. And so AI and ET, artificial insemination and embryo transfer, relatively old, but revolutionary in certain industries. Like 95% of dairy cows out there are the result of artificial breeding techniques. Moreover, sexing of semen is, scientists discovered ways, and I'll discuss these in the advanced session at noon, that we can take and ensure that our offspring are either male or female. Again, revolutionary in the dairy industry, where dairy males are not worthless, but really close to it in the eyes of most producers. So if we can increase the number of females, then we can increase dairy productivity immensely. And then this marker-assisted selection. Many of our elite purebred people may know what this is, having never been called marker-assisted selection, but when we send off a blood or a tissue sample to look for a genetic disease so that we can eliminate a male or female from our herd or flock, that's sort of marker-assisted selection. Likewise, we can use it on the positive end of the spectrum. If we know that there's a gene that increases marbling in cattle, we can test a 1-day-old bull calf and find out if he's worth keeping or not. And more details on that during the 12 noon session. So, animal biotech includes disease detection and mediation, of course, genetic testing, ELISA tests, Um, Many of you that are horse enthusiasts have to have Coggins test to transport your horse anywhere. That is one of the first examples of a commercial use of an ELISA test. The use of stem cell therapies nowadays in companion animals and in performance animals like horses is really starting to take off. One of my students whose mother is a veterinarian is starting to engage in stem cell therapy and stem cell therapy consults to help repair and mediate animals with issues. And biologicals, relatively new on the market. If you've watched human medicine commercials, which seem to be everywhere nowadays, you see all of the medicines ending in UMAB. Humira is an example of one of those. That's the commercial name, but it is a UMAB medicine. That's a biological, and we're actually using them in animals. And I've had the privilege to work on a couple of research studies with the swine operation, testing biologicals for the porcine epidemic diarrhea that knocked out a lot of pigs in Iowa. And so those aren't medicines in the sense of what we think of, but rather things that are helping the body combat the problem. And of course, then we do get in genetic modification. We learned about Dolly in 1996, scared the world to death. Not long after that, we started doing cloning here in the United States. Virginia Tech and Penn State have been involved in cloning trials with pigs and other animals to help develop agnostic organs. So organs that will be transplanted with less risk of rejection. And of course, if you do pay attention a little bit to the news, about a year ago, the USDA approved the first food animal GMO, which is genetically modified salmon that grows nearly twice as fast as its wild counterpart using the same inputs. And so that's some of the directions we go with those in animals. So what are the issues? And as a beginning biotechnology person, this is probably, if we go back to our original objective for the lesson, understanding the interface of society and science. And the issues is probably something that you would discuss in biotech, particularly with an introductory group of students before you would get into this, the how-tos of biotech. And this also, from an FFA standpoint, is awesome fodder for speeches because biotech is current, it's modern, it's technical, and it's sort of, I think, has a nice bit of wow factor when students can talk about the issues. So yeah, prepared public speaking and ag issues forum are loaded right here. So anyway, plant versus animals represents usually a pretty big issue. You don't hear of too many people expressing concern over playing with plants in a lab. But animals, and we've seen it, how often are animals used in testing that it really engenders a lot of human emotions. So you generally will find a lot more emotional response in animal-related biotech than plant, until you start talking about plants for food. And then people start to make plant biotech a big issue because they don't want their children to develop extra body parts or cancers or whatever has been promulgated through social media as a result of biotechnology. Other issues is the transition from molecular biotech, from macro to molecular biotech. No one cared that we were breeding, say, for example, a dairy bull to a beef heifer or vice versa, because that's like normal and that's kind of old. It's thousands of years old. And people can see that happen. Even plant breeding, if you take one strain of wheat into another greenhouse with a different strain of wheat and you tap the heads together to cross-pollinate, hoping to develop a new third strain of wheat due to some crossbreeding, no one gets upset about that. But when you get into molecular biotech and you say, oh, well, we're going to shut down this one gene that causes this animal disease so the animals are healthier, they can't see that with their own eyes. Moreover, most of the world is not really familiar with molecular biotech. Biology, science, and math are not subjects that people are super comfortable with. They're not subjects that people in school often aspire to, and so their understanding of science is limited, and as a result then, they get scared of it. And so that creates issues. They don't remember A, C, T, and G, and the fact that almost every organism on Earth uses the exact same 4 or 5 letters in DNA, and that if we switch them around a little bit, it's really not making any chemical change of significance. But because they lack that understanding, and they didn't use it a lot too. I mean, a lot of people don't use the understanding of genetics, what I call the central dogma of molecular biology and protein synthesis. So because they maybe did that in 10th or 11th grade in high school, and if they didn't take a scientific discipline in college, they're like lost. And as a result, they don't understand, they're scared, and fear leads to controversy. And then also demographic characteristics lead to issues. So religious background and upbringing. I had one of my very best students ever, really, really top end performer, but she was a very devout Catholic, and the Catholic religion does not agree with a lot of biotechnological initiatives and procedures. And so she was opposed to that. It was awesome that she was willing to open her mind and learn about them, because that makes her a much more complete opponent, in my opinion, than someone who just hates it based on the fact that they don't like it. Cultural heritage, education level. We note, and a lot of my students who do science research at the social level have studied this, and we find a pattern that people with higher education levels seem to be a little more amenable to biotechnological advancements. And I think it goes with the fact that they probably have studied this DNA and genetics of organisms a bit deeper. And finally, proximity to agriculture production sequence. Agriculturalists seem to be more willing to accept biotech because it makes them earn money better. It or widens their profit margin in some cases, or diminishes their input costs. And so you find that people that are farther and farther away from production, processing, and even marketing of the ag products. So as you move away from producer and more towards the consumer end of the continuum, you see issues become more prominent. So, okay. So those are the general issues. What are some of the specifics? Antibiotic use and resistance is big. In fact, we've heard that a bunch in the last how many years, overuse of antibiotics in farm and agricultural animals has led to contamination of water and creation of superbugs. There is some science behind that, but it's not real strong, but people believe it because it was on Facebook. And so what we've seen here is a lot of advertisements on TV catering to that consumer trend to want no antibiotics. And I didn't even put hormones on here either, I love the one soup company commercials where they say it's now hormone and antibiotic free. Well, hormones have never been approved in poultry production, period. But people wouldn't believe that, so they say we've stopped. It's easier to be a rehabilitated addict than it is to say you've never used it at all for some reason. Cloning creates issue. I'm sure you've dug into that. Your kids have asked. Bioremediation creates some issue, because what happens if this bacteria that we've modified to deal with an environmental problem escapes, crossbreeds with a wild bacteria, and starts creating bacteria that destroy natural positive things of the environment? So that creates people and creates worries. Of course, farming is always an issue. What happens if my child eats a banana that has some type of female enhancing hormone into it? Is my son going to develop breasts or something like that? And so you see frankenfoods, they call some of these. Stem cells gets in there because of religious implications, and pulling stem cells out of embryonic animals, which requires interfering with the reproductive process, and in some cases taking the embryo out before term, which causes embryo never is the term. It turns into a fetus at some point, but it creates a heck of a lot of controversy, especially with those folks that are also against human reproductive technologies as well. And so abortion, people against abortion generally are the same group that generally tend to be against stem cell therapy from embryonic stem cells. Reproductive technologies do create some issues, although artificial insemination, embryo transfer tend to be not as controversial because they've been around for so long. But when we start getting into embryo splitting and even gene therapy, on animals and plants in an embryonic level. Yeah. And then it trickles forward into the human aspect. If we can genetically test an embryo for diseases, does that mean we can also genetically test it for desirable characteristics? If I'm a parent with a lot of money and I want a designer child with blonde hair and blue eyes, can I test an embryo for those genetics? And if it does not have them, terminate the pregnancy and try again so that I can get the baby that I want because I have some, I hate to be this judgmental, but I'm superficial and I want my child to look a certain way, you know? So there's been a lot of that as it trickles forward into the human realm. And of course, genetic engineering and manipulation. Just because we can modify an organism doesn't mean we should. Is that playing God? So that's some of the big issues, and they might be things that if you're deciding to introduce biotech to a group of students, maybe you actually put them on that and say, hey guys, we're going to look about this and find out what does the public believe? What does your schoolmates believe? Do a survey of 100 students in your school and come back with what they know, what they don't know, what they believe, what they don't believe, what they agree with, what they don't agree with. And now we have good fodder for a really competitive, in my opinion, speech at an FFA competition. And so here's some of the classroom ideas, and I'm not going to click on each one of these links, but you can go into them yourself. Two different companies, Carolina Biological and Biotech, both produce an oil-eating microbe kit that is pretty sustaining. It's pretty complete. You don't need to have a lot of extra. And if you do need to add extras to these kits, it's like stuff you get at the grocery store. And so what the kids are doing here is they're investigating, do these bacteria clean up an oil spill efficiently? And so they develop a science research project. So these both here go really nice hand in hand with Ag Science Fair. Right? So you have a kid and they simulate a engine oil spill. Someone is changing oil in their car and they let it all fall out on the ground. They don't put the oil pan at the right spot. Can I clean it up before it contaminates groundwater? So they can sort of simulate that maybe in an aquarium. They bring in soil, they dump oil on it, and then they put these microbes on and they evaluate it. Both of these kits, I think, are, excuse me, both of these kits are relatively inexpensive. I believe that Carolina kit's around $100. The Biotech kit might be just a tick more. We use these kits in our biotech course as well as we start getting into application phases of biotech. We also can start testing for GMOs. And so this kit from EnviroLogics is actually not an educational kit, but it is a professional test kit for a purchaser in the field. So if I work for Kellogg's and I need to buy GMO negative soy and corn tissue, in other words, say for example, my cornflake suppliers didn't give me as much as I want and I'm in a shortage, I need to buy more cornflakes, and I'm going to be selling this as GMO-free cornflakes. I need to make sure that the corn I'm buying is not genetically modified. And one of the common genetically modifieds out there is Roundup readiness or glyphosate resistance. And so I can go out here and in a matter of about 15 minutes, stand at a producer's corn bin, take out a couple of kernels, crunch them up, and test them. And this kit right here tells me if it's modified for glyphosate resistance or not using bottled water and a small little what's called a lateral flow test strip. It works pretty much like a pregnancy test. You crunch up the corn tissue, you mix it with water, you let it solubilize for a couple of minutes, and then you put the quick stick in, and in 5 minutes or less, it's going to tell you whether it's modified or not. This kit is actually a little more expensive. It's got like 50 or 100 test strips in it, so it's something you might group the kids with, but it can be used to show them how we can test for GMOs or not. Obviously, making ethanol is something that you can get on the communities of practice and find all kinds of different things, but this is a kit from LabAids up there out of New York that gets you the various different scientific tools, such as a condenser unit and some of those things, so you can have the kids investigate making ethanol using different inputs, whether it's corn, honey, rice, other cereal grains, grass and green matter tissue. And then also there's a selective breeding simulation on this website. This is a PBS simulation that goes into selective breeding, as well as creating a GMO plant. And the GMO plant is a bit more technical. I'll talk a little bit more about that during the noon session. But this allows students to use a flash-enabled device to learn how plant breeders make plants. And so it's a little bit of an older technology, but I would suspect that many of our early agricultural students didn't know that plants actually sexually reproduce. And so that website there might be something you can do if you have enough computer access as another introductory lesson to biotechnology. Here's a couple of helpful websites. DNA from the Beginning is 70, I think, 75 short lessons. Probably this DNA from the Beginning is really good for you as a teacher to use if you're not really great with genetic theory, because it goes all the way from the discovery of cells and stuff and that they have a nucleus through Gregor Mendel and then into Watson, Crick, and Franklin, and it really goes through all the way now forward into human embryonic research, DNA sequencing and stuff. This is the DNA Learning Center, the second one, DNA LC, the DNA Learning Center. These two come from the same source, which is a Cold Spring Harbor Laboratory up in New England, which is a really, really good source of research and education. The DNA Learning Center is great for a teacher too, because it's got other educational resources on it. It tends to be a little higher end, but it's got some cool animations as well. So if you decide you want to show your kids how DNA transcription and translation works during protein synthesis, they've got a couple of really cool animations on there, and not like Animoto animations off of, but like really high-end animations put together by really technical scientific animators. So, and then this YouTube that I found the other day while I was putting this together is, I think, 7 minutes, maybe, I can't remember, but it's a really cool refresher also. So this YouTube kind of goes through the first bullet in a faster way. So if you do have a solid scientific upbringing and remember your science training in high school and college, the YouTube might refresh your memory a lot more so than going through the 70+ lessons at the DNA from the Beginning. So there you go. That's the end of that PowerPoint, which I've shared with your conference organizers, and I'm sure they can hopefully share that out to the Pennsylvania listserv or whatever means that the PAAE shares common documents. Maybe they'll just simply put them on the PAAE website. But I'm opening the floor now to questions. I see there's actually one that's popped up now. Okay, Holly. I structure my class, I'm a case purist, so I don't mess with the structure too much. The one thing that I do with my classes that's not in the biotech curriculum from case is incorporate that speech or debate. I don't think that any kid will ever go wrong by doing an oral communications piece. So when we get to Unit 1, Lesson 1.4, it's called ethical do's and don'ts, or ethical rights and wrong, which introduces all these controversial things. I have each kid do a speech where I let them group into threes to do the FFA ag issues for them. I use their rubrics wholesale right from the FFA website. But when I get down to my class, the case curriculum is really laboratory oriented. You can talk to Darla and some of the other ones there from PA who have taken the biotech course with case. When you get into probably the third week of the course, you're looking at 3, sometimes 4 days a week of labs, and in some cases 5. Our biotech lab in case really hits that box that I showed in an early slide, which is about career preparation, not consumer awareness and literacy. So it's very laboratory intense. Is it research based? There is some research activity. So I make them do the research for the speech. They also do research on something called agrobacterium tumefaciens, which is one of the biological methods of genetic transfer, which we'll talk about in the 12 o'clock session later today. But there's a lot of just simple labs. In fact, the spiraling component comes out immensely in this course because we do a lot of skill building before we actually do meaningful labs. So we have to teach the kids to pipette. And so we do a lot of pipette practicing before they actually do any labs where they're doing meaningful stuff. We learn about how to make gels for electrophoresis and how to run an electrophoresis machine before we ever put real DNA in it. And then we run DNA in it with the intent of doing nothing with it until we build another skill, which is using restriction enzymes to do restriction, fragment length polymorphism studies, and DNA fingerprinting later on. So I hope that answers your question. Yes, it is very laboratory intense because our focus in my curriculum is career development.
Thank you to those of you that have asked Aaron some questions here. Keep them coming if you have any. Again, you can raise hand and we can unmute you to ask live, or you may type them into the chat box or the Q&A pod.
And you know what? I did not give you guys my email address, so I'm going to add it to the screen here real quick. So let me share my screen once again so that you folks can see my email. This is my school email. There it is, adgeima@carrollk12.org. Feel free to shoot me emails if you have questions. I'm always willing to help as much as I can. And if I'm not able to answer your question, I contact my good buddy Jim in Arizona, who worked in biotech industry before becoming a teacher, and he's like an absolute genius. I'm disappointed that the Case Institute was canceled this summer because I was going to be my third or fourth time to work with Jim, and every time I work with him, I learn so much, and it enhances my teaching skills. So.
Aaron, we've had two questions posed in the Q&A pod from Sarah Beth and Jesse.
All right, so Sarah Beth, if you see me looking up, it's because I've got multiple screens, gang. Our biotech course is in my district is right in line with the CASE sequence. So if you go to caseforlearning.org, you can see that it is the third course in our sequence. If you're an animal or plant completer, they start off with an introduction to ag course. Most of my kids take animal science, which is called Principles of Agricultural Science - Animal in the CASE curriculum. And then they get to biotech. They should, I hope and pray that they've had high school biology before taking biotech. But our school system does biology as 11th grade for most students. And I sometimes get students that are taking my course concurrently with high school biology. And that does not present a huge problem. But it does sometimes present a little bit of a problem because we dive into DNA months before they really get into DNA studies in high school biology. What I find is I actually helped a biology teacher get better scores on their state tests because those kids are digging in at a deep level a little earlier on. And when the biology teacher gets to it, the kids have heard it a few times earlier. I'm not sure what other states are doing. New Jersey does biotech and case biotech pretty robustly. In fact, if I remember, New Jersey offered biology credit for students earning the introductory case course and the animal science or plant science course. So at the completion of the first two case credits, New Jersey awarded biology credit for the science students as well. And then they went on into biotech after that. So. Jesse Schaefer. Okay, so Jesse, it looks like, yeah, you're trying to see how can I incorporate a biotech unit or how can I talk about this? And it's in natural resources and environmental just as easy as in domestic plants and animals. I think one of the things I might do with the kids to uh, get them aware is talk a little bit about the controversy and why is it something that's on the news a bunch. To do biotechnology justice, you have to spend a lot of time talking about and reviewing biology. And I don't know that in those a hort class and animal science class, you're going to have as much time to get into understanding the central dogma of molecular biology, which is DNA translates into RNA, which translates into proteins, and then proteins go do most of the stuff in the body or in the organism. It takes quite a bit of time, and you might not have. So I would encourage maybe talking about the issues. And as a student gets excited about a specific issue, they might dig into the how then. Um, when I have my students do the speeches, we talk about it's going to be a speech on a controversy, but before you talk about the controversy, you might want to talk a little bit about the simplicity of the science. So, before they talk about embryo transfer and why people like it and why people hate it, they probably need to explain what is embryo transfer. So that helps the students dip into that a little bit. If you check out some of the resources that I gave you also, Jesse, some of those might be cool little snippets that will help maybe expose the kids to biotechnology. Um, and maybe could then entice them to an independent study or maybe an agri-science research project. So I hope that's,
Jesse,
go ahead.
another suggestion I have is to maybe address it from the standpoint of the implications of genetically modified organisms, plant or animal, that might be released into the natural environment and the impacts on natural ecosystems and ecology that they may have. Just another angle you could take on that.
Yeah, genetic drift is what some scientists call that, when genes from a domestic organism make it into the wild nature. And so that's one of the controversies with especially crops, is the Roundup resistant or the glyphosate resistance gene migrating out of agricultural crops into wild relatives, causing glyphosate to become decreasingly effective and increasingly dangerous in the environment. So.
Well, folks, we are 3 minutes past the allotted time. I don't want to speak for Aaron, but I'm sure he would be willing to hang on and speak
Yes,
to
I would.
anybody individually. But we are scheduled to have a brief break in between this session and the hot topics session coming up next at 11. Aaron did a fantastic job. As you'll hear tomorrow, a lot of the key words and points that Aaron has raised are addressed in either a singular deep dive workshop that the center has planned for the coming year, or will be addressed in one way. So Aaron did a fantastic job of sort of segueing into the release of our center professional development program for the coming year, and did a really great job of sort of overviewing the biotechnology space, as well as giving you some take-home, boots on the ground ideas and resources to implement in your classroom. So with that, one final call for questions for the good of the order. Either again, raise hand, chat box, or Q&A pod. And hearing none, I'd like to thank Aaron on behalf of everybody, and he will be back at noon. Thank you all for joining. Hopefully see you at 11.